All right, folks, we're going to go over our second set of practice problems. So this is non-Mendelian genetics. We're going to start with incomplete dominance. So as it sounds, it's incomplete dominance. So we're going to use two different letters because neither is completely dominant. For example, in radishes, the gene that controls color exhibits incomplete dominance. Pure breeding red radishes crossed with pure breeding white. So pure breeding also means the same as homozygous. And these two together make purple radishes. What are the genotype and phenotype ratios when you cross a purple with a white radish? So here you'll see I made a key, W for white, R for red. So W then is purple. That's our heterozygote. So pure breeding white with purple. So purple is RW. So RW, RW, WW, WW. So genotype, again, those are the letters, the genes. So we get 2 out of 4, that would be purple, and 2 out of 4 would be white, or we could condense that into a 1-to-1 ratio. For phenotype, 50% are purple, that's our RW, and 50% are white. The second example is talking about cattle that also show incomplete dominance in the color of their coats. Pure breeding red, so we're going to use an R for red, and pure breeding white, a W for white, gives us a roan cow. So that's our heterozygote. So we're going to do RW for roan. So we cross 2 roan individuals, RW, RW. So remember, when we cross 2 heterozygotes, we get that 1-to-2-to-1 ratio for our genotype. And then we get 25% red here, 50% roan, our heterozygote, and 25% white. Codominance is what it sounds like. So like co-chairs, that is, both are equally dominant. We've seen this before with our blood typing. So we use a letter. So in blood typing, we use the letter I. So capital I is for a dominant and lowercase I is for recessive. The only recessive in blood typing is O. So we have codominance in blood type A and B. So when we cross those two together, we get AB blood type. If we have an allele for A and an allele for O, we only see the A because A is dominant. Same thing with B. We cross an allele with B and an O, we get blood type B. When we get 2 little I alleles, so those are both for O, that's the only time we'll see blood type O, as it is recessive. When we cross an A and a B, we get that AB blood type. Here we're crossing an individual who's heterozygous for B blood, because remember, you can also be homozygous. You could have big IB and big IB, or big IB and little I. Both of those will show only the dominant blood type, which would be B. So they are heterozygous. So that is big IB, little I, with an individual with AB blood type. So here we get big IA, big IB, so blood type AB, big IB, blood type B, big IA, little I, blood type A, big IB, little I, blood type B. So our genotype, 1/4 or 25% big IA, big IB, 25% big IB, 25% big IA, little I, and 25% big IB, little I. Our phenotypic ratio is going to be different because, again, remember, if I have big IB, or big IB, little I, I'm still showing only that blood type for B. So blood type for B is 50%, while A is 25% and AB is 25%. All right, so again, we're just looking at blood types and probabilities here. And one last example, we have sex-linked. The lethal dominant is a little different here. We'll get back to this in a second because that is a complex problem, not common on the AP exam. Sex-linked, however, is. When we see a sex-linked trait, it's found on the X chromosome more often than the Y, and we use those letters. So big X is female, big X, big Y is male, because females have 2 X chromosomes, males have 1 and a Y chromosome. So when we do this, we use that letter, we use the X and the Y, and then we put a superscript for our trait. So big H will be normal, little h will be hemophiliac. So the genes for hemophilia are located on the X chromosome. It is a recessive disorder, meaning the only way you can get it is if you do not possess a big H. The question asks us to list the possible genotypes and phenotypes of the children from a man who is normal and a woman who is a carrier. Remember, that's here, the carrier. She shows a normal phenotype, but she carries that recessive allele. So our carrier is big X, big H, big X, little h. Up here, we have X, big H, and Y. We cross these, we get a woman who is homozygous for the normal allele. We get a woman who is a carrier. She's heterozygous. We get a male who is normal. And then we get a male who does not have the big H. So therefore, he displays the recessive trait. He will be hemophiliac. Remember, in sex-linked traits carried on the X chromosome, males are more likely to be affected than females. You can see that genotypic ratio here and the phenotypic ratio here. It's going to be a little different because we're looking at the exact letters. So 25% X big H, X big H, 25% X big H, X little h, 25% X big H Y, 25% X little hy. 100% will be normal female, or you could say 25% homozygous normal, 25% carrier, if you would prefer. 50% normal male, and a 50% affected hemophiliac male. So sex-linked usually means X-linked unless they specify it's on that Y chromosome. Up here for lethal dominant, achondroplasia, which causes dwarfism, is a dominant trait. So if you have that large letter, you will have achondroplasia. If you have 2 small letters, you will be normal. Remember, you can only see that recessive trait if you have 2. If homozygous achondroplasia, so that would be homozygous, big A, results in death. List the genotypes and phenotypes of all potential live offspring. So this is why this question is on here, and I chose this question to go over because you have to read carefully. So we know that we're not going to have homozygous achondroplasia, big A, being a viable embryo. So our genotype on here, when we list, we do our Punnett square. A woman and a man with dwarfism marry. We want to know what their offspring are going to look like. So big A, little a, big A, little a. We know that it has to be the heterozygous big A, little a, because we just said big A results in death of the embryo. So if 2 marry and mate, they would have to be the heterozygous. So this is one of the trickier parts of these questions that they might ask on the exam. Our Punnett square reveals big A, big A, little a, big A, little a, little a, little a. They're asking for potential live offspring. So our genotype ratio is 2:1. So 2 will be heterozygous and 1 will be homozygous recessive. Our phenotype, 2/3 will have dwarfism and 1/3 will be normal. All right, stay tuned for practice problems 3.